I'm a geneticist, and what I do is I study genetics and their function and their expression and how they play into how plants fully display their morphology, how they function in the field, things like that. Right now, I'm doing research for CBI, which is under the National Lab at Oak Ridge, and we are essentially trying to get a better transformation pipeline going for switchgrass callus, which is what this is, switchgrass, in order to better maintain, transform, and utilize switchgrass as a biofuel. The end goal being using it as a strong throughput for jet fuel, starting off with switchgrass, ending with jet fuel. We're trying to modify the switchgrass to be a better input for that. If you want to be a plant scientist or somebody that works in plant science, specifically with genetics, then one thing you're going to have to develop is good tissue culture skills. And by good tissue culture skills, I really emphasize the good stale techniques and stale practices that keep our samples, our experiments, even our standard calli and standard transformations, keeping them free from contamination. I'll show you what contamination looks like. This is switchgrass callus that has been modified genetically to express a anthocyanin pigment. The genes come from beets, and they were placed into this switchgrass callus in order to express this red color. Now, this red color is typically used for visual marker selection, things like that. But I'm showing you this because this is a relatively clean plate. You can see it at the top. There's some contamination, but I'm going to show you what a pretty well-contaminated plate looks like. Now, if your stale technique is lacking, or if something happens, I mean, it's pretty common that contamination happens even with the best stale practices because we live in a world full of microbes and bacteria and fungi. But here's what a contaminated plate looks like. You can see it's got that milky, gooey grossness to it, and it's almost on every single piece of calli. So a really good example of some contamination of what it can look like. What is the contaminant? No idea. I just know that it's different than this one. 35S Ruby, it's probably flipped because of the camera. That is the genetic selectable marker that was placed into these switchgrass callus. This at the top says MSD5B1. That stands for Mersicogene Scooge. D5B1 is two different hormones that are within the media to cause the callus to continue to grow. 2,4-D and BAP specifically. Callus has to be replated about every 2 weeks in order to continue to be healthy and continue to grow. This is normal, non-transformed switchgrass callus, and again, callus is undifferentiated plant tissues and plant cells that can then be manipulated to do different things like shooting and rooting. They can be transgenically modified, like this switchgrass callus. But these are just typical switchgrass callus stock that I'm going to be switching over to new MSD5B1 media in order to maintain them for future experiments. The reason contamination is such an issue is because not only is it literally infecting plant cells, getting them sick and killing them, but it's also directly competing with any transformants, any plant cells, anything that we have going on in our petri dish. This is throwing a wrench into it. So it's important that we don't have contamination at all, but when we do have contamination, we either completely throw out the plate or recover it using our stale practices. When I bring up stale practices, I really mean working in a flow hood, wearing gloves, and a whole lot of ethanol. Spraying down 70% ethanol on everything, making sure we flame and spray down and submerge this into ethanol as well, and then drying the ethanol, which dries very quickly under room temperature, but also extremely quickly under the Bunsen burner. And we use these stale practices in order to transfer the callus as close as we can to this part of the flow hood. A flow hood, if you couldn't tell by the name, is shooting out a flow of passive circulating air, and it's doing this through a HEPA filter, which means it is pushing air that is completely sterile over everything. And essentially, the function is to create a zone of interference for the bacterial, fungal, or viral pathogens in order to keep them from landing on our petri dishes. Um, Combining that with the sterile techniques of ethanol and gloves and using sterile equipment like this. This is our pack of our new MST5B1 media. I created this pack by synthesizing the MST5B1 in a liter, autoclaving it, and then solidifying it using a solidifying gel into these petri dishes. I can do a video about that some other time when I'm doing more media prep. But essentially, these are completely sterile. They were removed from the walk-in freezer and brought into here and put into the flow hood where they will be opened in a sterile environment and used within a sterile environment. And you guys have already seen the transfer callus, so I'm not going to do that again. But I just wanted to touch on contamination and some of the things that are involved in sterile practices, so you can get a good idea of what a day in the life of a geneticist is like.